This time I'm planning on making a video on general bacteriology, but as I never made notes of general bacteriology on normal basis, I thought of using the questions that are mostly asked from these chapters. So I think all of us know that there's a book about on microbiology that most of us use, and the hint for that book is I'll give the hint right here. The hint is this. Okay, so it's something lip or lipin. Okay, so that is the book, and that is the book that where I'm going to use the book that I'm going to use to make the notes from. Not the notes really, but the questions. So yeah, so this is a question from chapter 2 that's very commonly asked. The cell walls. There are two ways to answer this question. The first way is that you can go to table 2 and you can see the answer from there. The 3 points, there are only 3 points given at in that table, and I think the examiner will be happy because you answered from the book exactly. Now the other way to answer that question is that you can make a table of your own. You can start with the property or the component that you're going to talk about. Then you can go with gram-positive bacteria and then the gram-negative bacteria. So the first property that I'm going to talk about, and then you'll write the property over here and then you'll talk about both of these. So the properties in that very table that is in the book are there peptidoglycan, the teichoic acids, and the third is the lipopolysaccharide. So we'll do those three first. So the first property is peptidoglycan, and the peptidoglycan is obviously thicker and it's multi-layered. In gram positive and in gram negative it's thinner and single layer. The next property, teichoic acids. The teichoic acid is present in gram positive and it's absent in gram negative. The next is lipopolysaccharide. It's a third property, it's endotoxin. It is not present in gram positive and it is present in gram negative. So the next property now is the gram reaction. This is not mentioned in our book and not in that very table. So yeah, so that is that gram reaction. For this is the gram for gram positive, it is. So the gram reaction is that it retains crystal violet dye, and for gram positive it is that it retains crystal violet dye and stain dark violet or purple, and they remain colored or they remain colored blue or purple with gram stain when washed with absolute alcohol and water. But the reaction for gram negative is, so about gram negative it is that it can be decolorized to accept counter dye, that is safranin or fuchsine, stained red or pink, and they don't retain the gram stain when washed with absolute alcohol and acetone. So now let's move on to the next property. We have done 4 properties already. So the next property is the periplasmic space. The periplasmic space is obviously absent in gram positive, but it is present in gram negative. The next is the outer membrane. The outer membrane is obviously absent in gram positive and it's present in gram negative. Then you can also mention mesosomes. More prominent in gram positive, less prominent in gram negative. Antibiotic resistance. Now the antibiotic resistance, it is more susceptible, the gram positive is more susceptible to antibiotics, and it is, whereas gram negative is more resistant to antibiotics. The next point can be that lipid and the lipoprotein content is low in gram positive and it's high in gram negative. Acid fast bacteria, you can actually write a little explanation for it, but acid fast bacteria have lipids linked to peptidoglycans, whereas this is high in gram negative due to the presence of an outer membrane. Next point can be flagellar structure. So the flagellar structure has two rings. The gram positive have two rings in basal body, but the gram negative has four rings in basal body. If the question actually you think that you need to prepare more, and because we are actually doing these to be able to prepare for the chapters, because general bacteriology near exams for revision, it's like you need to do a lot and you don't have time just to give to one chapter. So you can also do the diagrams of both of these, because that's also a question where it says that you have to draw the diagrams of gram negative and gram positive to differentiate between them. So you can also do that. Next question is about the medically important bacteria that cannot be seen in gram stain. Again, this question is from chapter 2. Only going to mention 4 of them, and I'm going to obviously you have to give, the question also states that you have to give reasons and the alternative approach that's taken when these bacteria cannot be seen in gram stain. So the first one is mycobacteria including M. tuberculosis. What is the reason and the alternative approach? The reason is that too much lipid in cell wall, so dye cannot penetrate. And then the alternative approach is acid fasting. Then we have Treponema pallidum. Treponema pallidum. The reason is that it's too thin to see. And the alternative approach is dark field microscopy or fluorescent antibody. The third medically important one we can say is chlamydia. The reason is that it's intracellular, which means that it's very small. And we can see it by inclusion bodies in cytoplasm. So chlamydia. Then there is next one is rickettsia. For rickettsia, the reasons are the same. And the other approach can be, the alternative approach for rickettsia is Giemsa or other tissue stains. So there was another question which is of importance from chapter 2, and it was what are plasmids and give its different types, which are the types of plasmids, and enumerate the structure and functions of function structure of medical importance whose genes are carried by plasmids. So we'll enumerate those over here and we'll just start writing the answers. So what are plasmids first? They are extra chromosomal, double-stranded, circular DNA molecules that are capable of replicating independently of the bacterial chromosome. Now, what are the types of plasmids? There are two types of plasmids: transmissible and non-transmissible. The transmissible plasmids can be transferred from one cell to another by conjugation. They are large since they contain about a dozen genes responsible for synthesis of sex pilus and the enzymes required for transfer. Non-transmissible plasmids are small since they do not contain transfer genes. They do not contain transfer genes. They are frequently present in many copies per cell. And for these, they are usually present in a few copies per cell. Now let's talk about the plasmids, the functions and structures, the following functions and structures of medical importance that are carried by plasmids. So plasmids carry genes for the following functions and structures of medical importance. The first one is antibiotic resistance, which is mediated by a variety of enzymes. Then there are the genes of resistance to heavy metals, such as mercury, the active component of some antiseptics. This is mercury is an active component, and silver, which is mediated by reductase enzyme. The third is resistance to ultraviolet light, which is mediated by DNA repair. The fourth is the pili. I don't know the correct the pronunciation for it, so they're also called fembrii, which mediate the adherence of bacteria to epithelial cells. And then there are exotoxins, including several enterotoxins. Okay, so now from chapter number 3, what is important is the growth cycle part, and in that growth cycle part there is a growth curve, and that the steps of the growth curve, those are the things that are important. So for now, I'm trying to draw this. I'm not good at it, but whatever. So over here, it's if it comes like draw the growth curve of bacteria. It's a common question. So that is log number of cells versus the time. Now let's talk about the phases. It goes on like this, and then it goes straight, and then it goes. This part is straight. I'm not able to draw it straight, but this is. It goes down like this. Okay, so I hope I'm able to draw it perfectly. This part is not this long. This part is a little less, so you can actually just make it lesser. I don't know if you see the diagram properly in the book as well. It will be help you. Now they asked us to label the diagram. So this part till here is the lag phase. You can call it A and write it over here that it's lag phase, or you can write it over there. It's up to you. Then this part from here till here is the log phase. Then is this part. This is the stationary phase. And this part over here is the death phase. This is the growth curve of a bacteria. So for this, you should know that during which this is the lag phase, during which vigorous metabolic activity occurs. But in this part, cells do not multiply. There's this metabolic activity. There's no multiplication of cells. So they do not divide, they do not multiply. So this can last for a few minutes up to many hours, this part. Then there is the log phase over here. In the log phase is when rapid cell division is occurring and the beta-lactam drugs such as penicillin act at this phase. Because during this phase, the drugs are effective and cells are making peptidoglycan that is when they're dividing. And the log phase is also known as the exponential phase. So come here, this is a stationary phase. Stationary phase is when nutrient depletion or toxic products cause growth to slow until number of new cells produced balances the number of cells that die, resulting in a steady state. Cells grown in a special apparatus called a chemostat into which fresh nutrients are added and from which waste products are removed continuously can remain in the log phase and do not enter the stationary phase. Stationary phase is only when the nutrient depletion occurs. That is what it says. Now come here at the T part. The final phase is the death phase, which is marked by a decline in the number of viable bacteria. So this is the death phase. So this is what you should be knowing about it from chapter 3. This was one important thing from chapter 3. Other things are also important if you want to read them, but this is what I found important and I think we must read it. There's another thing, aerobic and anaerobic growth in chapter 3. I think you should also give that a read. This is it for chapter 2 and 3. So we'll continue with chapter 4 after this.